The last problem we did in the last class before break was fractional precipitation. This is the problem where we had barium ion and strontium ion in solution. We calculated the amount of chromate that was required to precipitate each one, which is a type 4 problem. And we calculated the amount of barium left in solution when strontium started to precipitate. The question is whether we got rid of almost all the barium, and we did. We calculated that there would be only a very small percentage of the barium left, which means we got a good separation. I showed that if you won $1 million and the percentage was all you had left, you would only have $2.36. I also showed that if the next slide is 100% of the barium that we started with, here's the 100%. The next slide shows the amount that would be left if it was 0.000236%. And here's the amount left, if you can find it. This is a precipitation problem in which you do not multiply by the coefficients. So in the next problem, I would like to use an example where the compounds that are precipitating have a coefficient in order to demonstrate that you don't multiply. In this precipitation, we're going to separate magnesium from lead by using fluoride. I showed that as you add the fluoride slowly, you will begin to precipitate magnesium first, because magnesium fluoride has a smaller Ksp than lead fluoride. And you have to stop at the place where you would start precipitating lead. And so we'll then ask how much magnesium is left in solution when you get to the point where lead starts to precipitate. You can look up the Ksps of the two compounds in Table 1. And the first question is to ask what concentration of fluoride is needed to precipitate magnesium. This is a problem where you're going to set Q equal to Ksp, but you do not put a 2x in the bracket when you solve for F minus, because this is a mixing problem. That's where the number for magnesium comes from in the picture in the next slide. Now we have to do the same problem for lead. You repeat the same process for lead using the lead Ksp, and again, you do not put in the 2x in the bracket, and also lead has a different concentration starting than magnesium does, so you also plug in the starting concentration. That's where the number for lead comes from in the next slide. Now we want to find out how much magnesium is left in solution when lead starts to precipitate. Of course, magnesium precipitates first because magnesium fluoride has a smaller Ksp. The next question then is to calculate the concentration of magnesium left in solution when lead starts to precipitate, and that occurs at the larger amount of F minus. You have to use the Ksp for magnesium fluoride because you're solving for magnesium, but you also have to plug in the F minus concentration that corresponded to lead because that was the one that was the largest. This picture shows what the question part D is actually asking. Again, because this is a mixing problem, you also do not multiply the concentration of fluoride by 2 when you plug it in to calculate magnesium. This slide shows the amount of magnesium ion left, but we want to calculate it as a percentage. The percent left is larger than in the last problem, but it's still very small. It amounts to about 1 left out of 2,000 that you started with, or 0.05%. To use the $1 million example, in this case, you would still have $500 left, but that's still pretty much gone compared to $1 million. Again, if the next slide is 100%, here's the 100% again. The next slide is the amount that would be left in this problem. This is a little bit more than last time, but if you spread that square out over the entire screen, you wouldn't even be able to see it. So this is a good separation. The sixth kind of problem that I want to show is actually one called common ion problems. But this is actually part of another topic. I'm going to call it altering the solubility of insoluble salts. If you put an insoluble salt into water, of course it doesn't dissolve very much, but you can put 3 things into the water that will change the solubility. Sometimes the salt is too soluble and you can't get a complete precipitate. In that case, you can put in a common ion and it will decrease the solubility, so you get a better precipitate. The next thing you can do is, if you want to dissolve the precipitate, which you sometimes do in the lab, you can add a strong acid to the solution, and sometimes that works to dissolve it, and sometimes that doesn't. The third thing that you can do is to add a ligand to the solution, which forms a metal ion complex with the metal ion. We didn't talk about that yet, so I'm going to talk about that third one more in the next class. So we're just going to talk about 1 and 2 today. And the first kind of a problem that we're going to look at is a mathematical one where you add a common ion. I'm going to demonstrate this problem by first calculating the solubility of the insoluble salt, lead fluoride, in pure water. Then we're going to add a common ion to the water and calculate the solubility in the common ion and show that it will be less. What a common ion is that you could add to the solution either of the 2 ions that are in the salt. You can either add lead or fluoride to the water, but you don't want to add both because you don't want to make more solid. What you want to do is get the ions that are in the solution to come out as a solid. So the question is, what happens if you increase the concentration of either one? If you increase the amount of either the lead ion or the fluoride ion, you'll shift away from those by Le Chatelier's principle, and since you're shifting towards the solid, the salt will become less soluble. I first want to calculate the solubility of lead fluoride in pure water, and this is a type 2 problem which we've already done. I set up the ice table and the Ksp expression, and we then need to substitute the Ksp and the x's into the equation and solve. I look up the Ksp of lead fluoride in table 1, and we substitute the Ksp and the x's just like in a type 2 problem from last class. You can expand the side with the x's and divide by 4 on each side, and then take the cube root, multiply by the molar mass, and that will give you the solubility in grams per liter. 1.38 grams per liter is quite a bit, and so you might want to decrease that amount in order to get a complete precipitate. You can turn this problem into a common ion problem by asking what the solubility of lead fluoride is not in pure water, but in a 0.1 molar sodium fluoride solution. The sodium will act as a spectator, but the F- will act as a common ion because it will be common to both the solution and the precipitate. You expect the fluoride to decrease the solubility of the solid, and it changes the ice table because now there's not 0 fluoride at the beginning, but you added the 0.1, and you don't multiply that by 2 because the person doing the experiment is the one who chose the concentration. As you can see in the ice table, the last row for fluoride now contains 0.1 plus 2x, but we can ignore the 2x because that's the amount that comes from the solid, and that is going to be negligible compared to the original amount. You can cross out either plus or minus x, and that also includes 2x or 3x because those are still very small numbers. Since the 0.1 just dropped down to the bottom row, what you plug in when you do this problem is you plug in not the 2x but the 0.1 for fluoride and the Ksp on the other side. You can then divide both sides by 0.1 squared and multiply by the molar mass in order to get the solubility in the common ion. The solubility went down a lot. It went all the way from 1 gram all the way down to 0.01. If you want to know how many times less soluble, it is taking the solubility in pure water divided by the solubility in the common ion, and you find out that it becomes 79 times less soluble, which in practice is a lot, and I'm going to show you a picture of that in a minute. When you do these problems, you don't actually have to write the ice table if you know what to do, because as I showed here, if you start by setting up the pure water case with the x's in both parentheses, all you have to do when you do a common ion problem is to take the amount of the common ion, cross out what's in the parenthesis for that ion, and plug in the concentration that you're given, and then solve for x. This picture, which I actually did in the lab, shows that if you take the salt calcium sulfate, which dissolves pretty well in water, 1.6 grams per liter, but then you add to the solution 0.8 molar calcium ion as a common ion, you can see that everything that was in the solution precipitates because the common ion decreases the solubility. I want to do one of these problems where we start with the solubility of a salt in pure water and then calculate the solubility in each of the 2 common ions, because then you get every possible combination of substituting. We're going to use silver carbonate, and I'm going to show you the ice tables and how they work. But I'm also going to show you that you really don't have to use these, because if you know how to substitute, all you have to do is just do a substitution. When you do the solubility in pure water, as it was in the previous slide, there's x's that drop down for both ions. But if you do the solubility in 0.2 molar carbonate, the 0.2 just drops right down to the bottom row. And if you do it in 0.5 molar silver, the 0.5 drops down to the bottom row, because the x's are going to cancel relative to those larger amounts. Here's the Ksp of silver carbonate in table 1. If you calculate the solubility in pure water, you're going to put x's in each of the blanks, and then when you solve, you take the cube root, and x is the solubility in moles per liter. I'm not going to convert it to grams per liter this time because it depends on what you're asked for in the problem. When you're asked for the solubility in 0.2 molar carbonate, you don't really need to use the ice table. If you start with the pure water case and then find the parenthesis that corresponds to carbonate, cross out whatever is inside of it, and plug in the amount you're given. You can leave the other parenthesis alone, and then if you solve for x, you'll get the new solubility. When you expand it this time, the 2x quantity squared comes out to be 4x squared, and you can then complete the problem by solving for x. So I multiply 4 times 0.2, divide both sides by 0.8, take the square root, and x gives you the solubility in the common ion. If you divide the solubility in pure water divided by the solubility in the common ion, you find out that silver carbonate is 40 times less soluble in the common ion than in pure water. 40 times is not as big as 79 times, but it's still a significantly less solubility. Now, if you want to calculate the solubility in the other ion, 0.5 molar silver, you are going to take the pure water case with both parentheses having x's and completely cross out what's in the silver parenthesis and plug in the 0.5. You cross it out completely because the 2x is negligible compared to 0.5. You can then divide both sides by 0.5 squared, and you see that x is very less soluble. And if you take the pure water divided by the solubility in silver ion, it's 3.9 million times less soluble, which is obviously significantly less. If you look at this summary, you don't have to do all of the ice tables in order to do this problem. If you set up the pure water case first, cross out what's in the parenthesis, and plug in the correct number that was given, you can solve for x and get the answer by the method that's shown. And then you don't have to worry about the ice table and any other problems. You can just solve for x. I'm showing that list of 3 changes again because the next one we want to do is adding a strong acid. And I'm adding not only the solubility of the insoluble salts in a strong acid, but I also want to add strong bases because they act exactly the same way as insoluble salts do, and I'll show that. Instead of wanting to decrease the solubility of lead fluoride this time by adding a common ion, I want to increase it so that it dissolves by adding an acid, a strong acid, and I want to see whether this is going to work. So the first question is the H plus that you add able to react with either one of the ions that's already in the solution? Because the F minus is on the right of the arrows in the Ka table, that means that the F minus is a base, and it should be able to react with the H plus that you're adding. This is actually a type 3b neutralization, strong acid combining with a negative weak base. And when that happens, you are going to get an increase in solubility. Adding the H plus is actually like eating the F minus, and when you decrease the concentration, according to Le Chatelier's principle, you shift towards what you removed, and so the salt will become more soluble. This slide shows that an insoluble base, NaOH2, works exactly the same way, because OH likes to react with H plus even more than a weak base, because OH is a strong base, and therefore the effect of acid on a base is even greater to increase the solubility. This is one of the steps in the group 3 flow chart, which is the experiment that you're going to be doing online soon. And if you look at the main precipitate, it has 5 precipitated ions, but then you add a sulfate buffer, which is very acidic. That's pretty much like adding H plus. And if you look at it, it actually dissolves 3 of the ions, but not the other 2. And this is the main separation step. The equation at the bottom of the page shows that what's happening is that the H plus is reacting with OH to make the Cr3 plus soluble. And that's actually the dissolving equation for chromium that you write in your lab report when you turn it in. This is another example from the group 3 flow chart. When you dissolve FeOH3, which is actually rust, and you add sulfuric acid, you separate the iron from manganese, and you do it by the equation at the bottom of the page, which is exactly the same as the one for CrOH3, except this time it's the iron that gets released into solution. So it works exactly the same way for iron. The process of an acid dissolving an insoluble salt is really all or nothing. The acid either works to dissolve it or it doesn't. The problem is it doesn't always work. And I want to show you an example where it doesn't. If you try to dissolve the insoluble salt silver chloride by using an acid, you will not get any increase in solubility. So the question is why? The reason is that because of the fact that Cl- comes from a strong acid, if the H plus tries to react with it, it can't stick to it because HCl is always 100% ions. So if the negative ion in the salt comes from one of the strong acids instead of a weak acid, the acid will not have any effect on the solubility. The principle is that you can only increase the solubility of an insoluble salt with an acid if the negative ion is a base. And it could be one of 2 types of bases. It could either be a weak base, which is the conjugate base of a weak acid, and you would find that in table 2, the Ka table, or if the negative ion is OH, that will also increase the solubility in an acid. Here's an example that I did from the lab. I tried to dissolve calcium oxalate by adding a strong acid to the solution, and you would expect that to work because oxalate really is a weak base. You can see that it's a weak base because it's on the right of the arrows in the second step for oxalic acid in the Ka table. So in this picture you can see that calcium oxalate is completely insoluble in water. But when I added enough HCl so that the concentration was 3 molar, it dissolved completely. This is the way strong acid is able to dissolve insoluble salts if they're the appropriate kind. This is a summary, and I think this is another way to say it, that H plus will dissolve an insoluble compound if the negative ion either comes from a weak acid, which makes it a weak base, or the negative ion is OH. H plus will not dissolve an insoluble salt if the negative ion comes from a strong acid. So I would like to now do some examples to apply these and see how it works. This is the kind of problem I ask. All I do is say, will the following be more soluble in acid than in pure water or not? So the only thing you have to look at in BiI3 is the negative ion. And since I minus comes from a strong acid, it will not increase in solubility. For FeS, the only thing you need to look at is the S2 minus negative ion, which is a base because it's on the right of the arrows in the second step for H2S. So FeS is more soluble in strong acid. FeS is one of the compounds that precipitates in the group 3 flow chart, and the very acidic sulfate buffer does cause it to dissolve, as you can see on the flow chart. AlOH3 also dissolves more in acid, because OH reacts with H plus even better than weak bases. For calcium phosphate, we need to look at the PO43- ion. Since phosphate is on the right of the arrows in the third step of phosphoric acid, that means that it is a weak base. And so calcium phosphate will be more soluble in a strong acid. The next major topic we're going to talk about is metal ion complexes, which are also called coordination compounds. These are a few examples of them that are from the lab you do in group 3 and also group 2. I want to describe some of these and show you how important they are in chemistry, because if you're going into a medically related field, there's no question that you will see this again. So we have to try to give somewhat of an appreciation of them right now. If you look at the group 3 flowchart, 3 of the final tests in that group are actually metal ion complexes. The blood red iron complex, the yellow cobalt complex, and the strawberry red nickel complex. The final test for copper in group 2 is also a metal ion complex that is a deep blue solution. That's the positive test for copper. If you look at these complexes, the first thing you notice is that they're very colorful, as most complexes are. They can either be ions that are dissolved in water, or they can be standalone compounds that are precipitates, like the nickel DMG complex. You can also see that the bonding must be different from ionic compounds. If you were going to make a normal compound between iron and the ion thiocyanate, you would generally cross and get FeSCN3, but that's not what you get. And if you look at the copper complex at the bottom, what's attached to it is ammonia, which is not even an ion, it's a neutral compound. And so we have to talk about how all this works and how the bonding is different. I made this slide to show how important complexes are in science and medicine. You are going to deal with them again if you're a chemistry or biochemistry major, because about 80% of the class called Advanced Inorganic Chemistry, which you take following this one, is about complexes. Obviously, I cannot give a complete undergraduate course, so I'm going to try to give you the most important things about complexes that are related to medicine. Most colors in nature are either metal ion complexes or organic dye molecules. Sometimes they're bonded together. These are things like leaves and fruit and bird feathers and anything else that's colorful. Both chlorophyll, which is what collects light from the sun for plants to make food, and heme, which is what carries oxygen in the blood in order for animals to be able to metabolize food, are both complexes. Most enzymes require at least one metal ion bonded in the structure to work. And metal ions are activators or inhibitors at a lot of points in metabolism, so they're very related to biochem, and they do it through complexes. The electron transport chain complexes have metal complexes in them, and that's how the electrons are transferred through the chain. And the DNA study is also involves metal ions in that they bind to DNA, especially the phosphate backbone, and turn genes on and off. So they're also involved in cancer research and genetic research of all different kinds. You can spend an entire career studying the binding of metal ions to DNA because of the importance of the topic. So we want to try to show enough about these so that when you take further classes or see them in biochem, you'll understand what they are, at least how they work and how they're put together. So let's look at the actual bonding in complexes. Let's say you were a good 131 student and your teacher said, what will happen if you put together the ions tin, 4, and chloride? If you were going to guess, this was probably going to be ionic, and more than likely you put the metal first and then cross, and you would get the formula SnCl4. The first problem with this is that ionic compounds are crystals, but SnCl4 exists, but it's a liquid at room temperature, which means it has to be covalent because it's not a crystal. I did say before that it's about 3% of metal compounds that actually are covalent. So the idea that a metal makes a compound ionic is about a 97% rule. And if you put tin 4 and chloride together in water, they don't even make SnCl4. They make a complex where 6 chlorines combine with 1 tin to make a -2 ion, which is a complex. But if you look at the charges, they do add up because +4 - 6 does equal -2. The first thing I want to tell you, though, is that you do not have to ever predict when a complex will form, because that's advanced inorganic chemistry and it's done experimentally mostly. When I give you a problem about a complex, I will give you both the formula and the charge and the fact that it's a complex and ask you questions about it. I wanted to show here that complexes have exactly the same kind of molecular geometries that you learned about in Chem 131. But there's only a few of them that are important in complexes, so you're not going to have to relearn them all. I'm going to show you the ones that are important. These structures were originally determined by a technique called X-ray crystallography, but now they can be done by computer modeling. The structure that you see there for SnCl6 shows that it's a square with 1 Cl up and 1 Cl down. This is a structure that's called in molecular geometry an octahedron. You remember words like tetrahedron and trigonal and square planar and bent and all these words that have to do with molecular geometry. We're going to look at those again. But this is the SnCl6 complex. Because the complexes are most of the time ions, you can make the same kind of compounds with them that you can with any other negative ion. And I showed 2 compounds here, sodium sulfate and then sodium connected to the -2 tin complex. So the formulas would be the same. The only difference is that the complexes are always put in square brackets in formulas to show that they are complexes. And the metal ion is always in the middle in the complexes that we study, so it's the central metal ion. And the things that are attached to it around the outside are called ligands. This is a Greek word that means grabbing onto something and then you become the partner of it. There's no comparable word in English. so they use the Greek word ligand. Now, what kind of bonding is there between the metal and the ligands? As I showed up above, the chloride has 4 unshared pairs, and one of them can form a bond with the metal. It's essentially a covalent bond, although it has some of what they call ionic character, but you can think of it as covalent. When you get a covalent bond where both electrons come from the same atom, that's called a coordinate covalent bond, and that's the kind of bonding in complexes. So that's the reason why they also call them coordination compounds, because that's the type of bonding. That also explains how a neutral molecule like ammonia, which is not even a negative ion, can bind to a metal, because all it has to have in order to bind is one unshared pair, which ammonia has on the nitrogen. So when ammonia binds to copper, there's actually 4 of them, and the figure that you see is what's called square planar. It's an octahedron without anything sticking up or down. And I showed the nitrogens as the atoms that are bonding to the copper. So a couple of the formulas look backwards, but the truth is that it's to show which atom binds to copper. And I showed the formula at the bottom. The ammonia is in parentheses, and you put the charge just like any other ion. Because of the fact that the copper complex is a positive ion, I can write a formula with any negatives, and the ones I chose were chlorides. So there is a compound like this, and it's neutral, and I put the copper complex in square brackets. One question I want to ask, though, is why did I put this one complex first? But in the sodium combined with the tin complex, I put the complex second. There's no mystery about this. In an ionic compound, the positive ion always gets written first in the formula. So if the complex is positive, it goes first. If it's negative, it goes second. Now right away we have to get into a type of ligand that's called a bidentate ligand, because they're so important in complexes. What I showed at the top of this slide is the structure of a molecule called ethylene diamine, which I abbreviate EN. You do not have to know these names, because you're going to get a table that you can look at in order to figure out everything you need to about ligands. But I wanted to show you that, as you can see, this is an organic molecule, but it's not that complicated. The 2 carbons have 4 bonds each, and each of the 2 carbons is connected to a nitrogen, and nitrogens have 3 bonds each. Now each of the nitrogens has an unshared pair. If you turn the molecule at 90 degrees and you rotate it, that's what I did below in the complex, and you can see that this complex has both nitrogens binding to the copper, and you can stick one of the ligands on each side. So they call the ligand bidentate because dentate means teeth, and it's like there's 2 teeth binding. And they also call these chelates because they also look like claws. They're trying to grab the copper like a claw. These bidentate ligands are very common, and ethylene diamine is usually the one that people see first because it's the most basic one. The table that you're going to get to look at all the time to do these problems with complexes is table 5. And if you look at the table, the EN that I just showed you is near the bottom where the green arrow is, and you don't have to know the name. I abbreviated EN, but what are the other things on this table? At the top, you see monodentate ligands. What that means is these are ligands that only bind at one place to the metal, like Cl-. And at the bottom is the class bidentate ligands, which bind at 2 places. Under those columns, I also show the charge of the ions. Under both monodentate and bidentate, some of them are neutral and some of them are anions. And if you look at both groups, the anions can have a charge of either -1 or -2. This means that if you're looking at an individual ligand, the number of binding sites is completely independent of the charge. So you have to know both. The number of binding sites is determined by how many arms the ligand has, sort of. But the charge is determined by how many electrons you need to make an octet. So if you know, for example, that a ligand is monodentate, and that's all you know, you cannot tell what the charge is unless you look it up, because monodentates can be either neutral, -1, or -2. And if you know that a ligand is bidentate, it's the same. You can't tell. Also, if you know that a ligand is -1, you can't tell whether it's mono or bidentate, because it still could be either one. So you have to look up and know both of them. The squawk that you heard in the previous slide was my son Conyer Wellington, who was not happy about something. That's the problem with living in a zoo. Anyway, you can have ligands that actually bind at more than 2 sites. I'm going to show you pictures of some of these, but you can have ligands that bind at 3, 4, or 6 sites, and even more in some cases. We're going to look at pictures later. This is a picture of another bidentate ligand called oxalate, as you're familiar with. Oxalate has 2 oxygens on it that can bind to a copper. The other 2 oxygens stick out the other side. So oxalate is also a bidentate ligand. But if you look at the formula of the complex at the bottom, it's listed as being a -2 charge, because all you have to do is add the charges. There's 2 -2 oxalates and a +2 copper. That adds up to -2. That's why that's the charge. In Table 5, oxalate is at the very bottom, and it's listed as bidentate and also as having a charge of -2. The names that you see there are unusual looking, but that's what they call these ions and molecules when they are in a complex. Believe it or not, this is actually the molecule heme that carries oxygen in your blood. That whole ring around the outside of the iron has no breaks in it. It's all one molecule, and it's called the porphyrin ring. The iron in the middle bonds to 4 nitrogens, and they're in an exact square, so the iron fits right in. Heme is what's called, or the porphyrin ring around the outside then, is what's called a quadradentate ligand, because it binds to the iron at 4 different places. I'm not going to put anything larger than bidentate in your table, because these are all organic molecules, but if you get the hang of how to do the mono and bi, you can do any of the others also. If you look at the without the iron, it's all one ring around the outside called a porphyrin ring. And the other thing is that if you put in the middle a magnesium and change a few of the groups around the outside, now you have chlorophyll. And of course, when it's got iron in it, it's red. But when it's got magnesium in it, it is green. That's how much the colors can change. This is to give you a little bit of an idea of how biochemistry works. How is that heme unit connected in the protein hemoglobin that carries oxygen in your blood. If you can see, hemoglobin is a 4 subunit protein, and the subunits are arranged in a tetrahedron, and there's one heme in each. If you look at where the heme is attached, the protein has an amino acid residue on it called histidine, which has 2 nitrogens. One of those binds at the 5th position of the octahedron, and at the 6th position, oxygen can come on and off. So when you're in the lungs, the oxygen is not there, so it binds because there's a large concentration of it. When the hemoglobin carries the oxygen to the tissues, CO2 is coming out, which makes the blood slightly acidic, and it changes the conformation of the hemoglobin a little bit so that the oxygen can slip out and move into the tissues. Then the hemoglobin can return to the lungs and get more. This also shows how some of the poisons work. If you look at the molecule carbon monoxide, the carbon monoxide binds to heme at the same place as oxygen, but it binds much more tightly, so it blocks oxygen from binding. So as soon as you get a carbon monoxide on a heme, the oxygen can no longer be carried. So when you're breathing in carbon monoxide, you don't get any oxygen to the brain, and that's why it's toxic. If the heme gets a carbon monoxide bonded to it, you can't just pull it off. Your body has to break down the entire porphyrin ring and take out the iron and put it in a new ring. The thing about it is you've seen the colors of the heme ring, because every time you break one of the bonds, it changes color. If you've ever had a bruise, you know that bruises start out purple, and then they go through a red phase, and then a greenish phase, and finally they're yellow, and then they disappear. Each one of those colors is a breakdown product of the heme ring. And the final one is the yellow one called bilirubin. That's the one that's secreted into the blood and that the kidneys eliminate. That's the reason why urine is yellow. It's the breakdown product from the heme ring. This complex is an example of a hexadentate ligand. EDTA is a very common ligand that's used in analytical chemistry and also, for example, as a food preservative. The iron in the middle is right in the middle of that EDTA ring, and there's actually 6 atoms that bind to the iron, the ones that are shown in red. And each one of the rings is actually what's called in organic chemistry a 5-membered ring, because the iron is in the same ring as the parts of the molecule that are shown. The thing about this is you can take the iron out of it, as I'm going to do in the next slide, and show that the EDTA around the outside is all one molecule. This is what EDTA looks like at the top without the iron, and you can see that it's completely one molecule with no breaks. If you stretch it out, you can see what the structure is at the bottom of this slide. The part of the molecule that's in the middle is the 2 carbons and 2 nitrogens are ethylene diamine, and it has 4 acetic acid groups attached. That's the reason why they call it ethylene diamine tetraacetic acid, and that's what EDTA stands for. So when you put this molecule next to an iron, it wraps the iron up in an octahedral structure, but the molecule has 6 arms that bind, so it's called a hexadentate ligand. If you can understand these two concepts, coordination number and the number of ligands, we will be ready to do the next lecture where we talk about several of the properties of complexes. So I want to describe these. The coordination number of a complex is the number of attachment points or bonds to the metal ion. That is not always the same as the number of ligands, because a lot of the ligands bind at more than one spot. And I'm going to show you this with examples. The first complex on that last page is SnCl6. If you look up Cl in the ligand table, you can see that it's monodentate and that it also has a charge of -1. We showed this in a picture, which is an octahedron shape, before. So the first question is, what is the coordination number of this complex? Well, if you look at the tin, there are 6 bonds to it, and that is the coordination number. So the coordination number is the number of bonds that are sticking out of the metal. The number of ligands is the same, because each one of the Cl minuses is considered to be an individual separate ligand. So each one of them is monodentate and it binds at one site. So both the coordination number and the number of ligands is 6. The next complex we're going to look at is the copper combined with ammonia. So the first thing you have to do is look up ammonia in the table to find out what the properties are. You can see that ammonia is listed as monodentate and also neutral. We showed the structure of that complex before, and you can see that each one of the ammonias binds at one site. So the copper has 4 bonds attached, and it also has 4 separate ligands. Each one of the ammonias is a separate ligand. So the coordination number is 4, because there's 4 bonds to the copper. And there are also 4 ligands, because each one of the ammonias is a separate monodentate ligand. Now the next one, copper En2, is different because En is actually bidentate. So the first thing we need to do is look it up in the table. En is listed in the table as both neutral and bidentate, which means that it has 2 binding sites. We showed the picture of it before, where there are 2 ENs attached, 1 on each side. So if you're going to ask what is the coordination number, if you look at the copper, there are 4 bonds attached to it, so the coordination number is still considered to be 4. The coordination number is not dependent on how many ligands there are. It only depends on how many bonds are coming out of the metal. But this complex is only considered to have 2 ligands because the En on each side, each one of them is considered to be 1 ligand. So you have 2 ligands binding at 2 sites each. That gives a coordination number of 4. The same thing is true for the copper oxalate complex at the bottom. The charge is different, but you're going to see that the structure is very similar, and we already drew a picture of it, so we have to look up oxalate first. In the table, you can see that oxalate is listed under bidentate with a charge of -2, but because it's bidentate, it has 2 binding sites. This is the picture of the complex, and if you look at the copper again, it has 4 bonds to it. So again, the coordination number is 4. The next question is how many ligands? There's actually only 2 again, because each of the 2 oxalates is separate. So that gives you 2 ligands, 2 binding sites each, which gives a coordination number of 4. Now, even though I don't have any more than bidentate listed in the table, I do want to do the same problem with some of the bigger complexes I showed you. This one is impressive. The question I want to first ask is, what is the coordination number? If you look at the iron, there are 4 bonds sticking out of it. That's all you look at to tell you that the coordination number is 4. When you ask how many ligands there are, though, that entire porphyrin ring around the outside is all one molecule. This is one molecule with 4 binding sites, and so porphyrin is called a quadradentate ligand. So even though the coordination number is 4, there's 4 bonds, there's only one ligand, because the porphyrin ring contains all 4 nitrogens that bind. So that one ligand gives you 4 binding sites. The EDTA one is even crazier, because if you count the number of bonds to the iron, you're going to see that it's in the shape of an octahedron. So there's 6 bonds sticking out of the iron. That means that the coordination number is 6, but how many ligands is that? I showed you that the EDTA molecule actually is one individual molecule, but it has 6 binding sites. This is what's called a hexadentate ligand. So since that molecule is all one, that means there's only one ligand, but the coordination number can still be 6, because that one ligand or molecule gives you 6 different binding sites. This is the end of this class. In the next class, we're going to talk about more of the properties of complexes, and I'm going to give you about 7 different individual topics that are important for these, because I know that you will see them again in your career if you continue in chemistry, biochem, or medicine. So you can now do homework 9, and we will try to do the material for homework 10 and also introduce kinetics in the next presentation.